Hi everyone, I'm Professor Sarah Rankin and I'd like to welcome you once again to Imperial COVID-19 lockdown lessons. So today is lesson 3 and we're going to be hearing from Dr. Michela Noseda about how scientists are planning to investigate the genomic response to COVID-19. So please make sure you have Mentimeter ready on your mobile if you'd like to join the quiz. And feel free to post any questions you have in the chat and Margarita will pick them up. So just to tell you a little about Michela, she's a lecturer in the National Heart and Lung Institute here at Imperial. She normally does research into heart disease. And today she's going to tell you about how the cutting edge technologies that she's using to investigate the genomic signature of cardiac cells are now being applied to investigate the genomic response of different cells in the body to COVID-19. So let's go straight over to you, Michela.
Thank you very much, Sarah, and thanks everyone for tuning in today. It's so exciting, my first ever webinar. So I just want to start telling you that cardiovascular disease is the first cause of death worldwide. And ischemic heart disease, your heart attack, is alone the foremost global cause of death for men and women, despite reduction of risk factors. We all know we shouldn't smoke, we need to exercise and eat a healthy diet. So the high prevalence of myocardial infarction, of heart attacks, is related to the aging of the population and urbanization. People are moving from the countryside to the big cities. Now, today, I'm not going to talk about heart attacks and how the heart changes, but I want to focus on the technologies that we're using to study this amazing organ. A lot of my research focuses on the cellular landscape of the heart, both the healthy and the diseased heart. How can we study the heart? So classical experiments consist in taking sections of a portion of the heart, staining them and looking down at the microscope. You can see here in pink and purple some histological sections. In purple, you see the nuclei of the cells, and in pink, you see the cytoplasm. What can you tell from an experiment like this? Well, you can describe how the tissue is organized. You can probably see differences, you will see differences between a healthy and a diseased heart. If you're a little bit more adventurous, you can decide to dissociate the tissue and isolate the single cells. So for example, here I'm pointing out to a single cardiac muscle cell. Using mechanical and enzymatic digestion processes, you can release the cardiac muscle cells and all the other cells of the heart. Here there is a beautiful image of a single cardiac muscle cell. Look at the structure and these beautiful striations. Cardiac muscle cells are called technically a cardiomyocyte, and they're the functional unit of the heart. You've been seeing this video of how beautifully that cardiomyocyte was beating. It's essential that these cells are functional to maintain the heart pumping blood around the body. Now, the heart is not just made of cardiac muscle cells, so although these are fundamental, we also need specialized cells of the blood vessels. We need immune cells. The heart contains nervous system cells as well as fibroblasts. These are a crucial type of cells of which we don't know that much. We know that they're necessary to maintain the structure of the heart or any other organ. We know that they can increase a lot in certain pathological situations, but there's so much that we can learn more about this. Now, here is a crucial question that I would like you to think about. What makes these cells different one from another? I'm not going to give you an answer, but I'm going to give you 2 more questions to think about for a couple of seconds. Is DNA the same in all cell types of an individual? Just think for a second. And then I throw there another question. Is RNA the same in all cell types of an individual? So, sure that many of you have thought to the right answer. Within the cells of our body, all the cells have the same DNA. They have all the same genome. So what makes a cell of the brain, a neuron, different from a cell in the heart, a cardiac muscle cell? Well, there is an invisible machinery that activates certain sets of genes, and that pool of genes is then expressed, and you get synthesis of RNA. So active genes means RNA, and the set of genes expressed in the brain, in the neuron cells, is going to be very different from the set of genes expressed in the cardiac muscle cells. Let me tell you how what techniques we can use to analyze this set of active genes within specific cells. I'll make a couple of very practical examples because the concept can be a little bit difficult. So in a standard experiment, we look at the RNA, so the active genes within a tissue, in bulk. We say we do a bulk RNA-seq. Let's pretend we do an experiment. You guys are in your school assembly, and the experiment, each one of you represents a cell, and the experiment consists in understanding what color hair everyone has. In the bulk experiment, we'll take everyone's hair, put them together, and look at the color. I'm sure some of you have probably dyed their hair green like my daughter did, because you're in lockdown and you're allowed to do that. So will we see green hair in this bulk? We might, but we'll never know who was the one that dared to dye their hair green. If we see gray hair, we won't know that it comes from the head teacher. So this type of experiment will give us information, but we'll miss some details. Now we have single-cell genomics technologies that will allow us to look at the hair color in each single person. Let me give you another example. The bulk RNA sequencing or gene expression experiment is basically equivalent to looking at the smoothie. Now with single-cell technologies, we can look at each single fruit and know which gene is expressed in which cell type. This slide summarizes, we go down to the experimental setup, how we run these experiments. So we've got our tissue. We can dissociate this tissue and we'll have a number of cells. If you look down the microscope, what can you say? Well, you can look different morphologies, but you can't tell much about the function of these cells. Now we've got technologies that allow us to pick one cell at a time. Some technologies allow us to make a micro droplet and pick little cells. Then we get the RNA out of these cells and we sequence it. And I'm showing here an example of a code of the genome that in this case will be called transcriptome and will give us the results of what genes are active in that specific cell. The set of genes that are active in that specific cell are equivalent of an ID card. We know, which will tell us what the cells, their features, and we can make inferences of their function. Now I have a nice question for you. I'm very curious to know what your answer. You can use the Mentimeter now, and I'd like to know if you guys can estimate how many cells are in the human body. We have a beautiful technique, but we probably have a lot of cells to study here. Wow, I can see already some of you have tried to answer, and we gave you some options here to guess, and to see if our estimate, if you come out to an estimate similar to what we have calculated. We'll just wait for a few more seconds to see if someone else dares to guess the number of cells in our body. So shall we just move on to the next slide? And I want to say that yes, most people know that it's a lot of cells, and the estimated number is 37 trillion. So there's a lot of cells that we need to study. So a lot of cells requires a big team of scientists. And two amazing scientists, Sarah Teichmann at the Sanger Institute at Cambridge and Aviv Regev at the Broad in Boston, have indeed funded the Human Cell Atlas. This is an incredible initiative that aims to create a comprehensive reference map of the types and properties of all human cells. This will give us the basis to understand health and disease, to understand tissues and organs in healthy conditions and disease conditions, and will give us input for generating new diagnostic tools and new diagnostic markers, as well as monitoring disease and identifying new therapeutic targets. The HCA, it's a true global community of people that want to work together. If you think teamwork, I haven't seen anything better than the HCA. It's a growing community. At the moment, there are almost 2,000 members. There are scientists from 71 countries, and there are over 1,000 institutes involved in aiming to create a human cell atlas. At the Human Cell Atlas, we don't just care of what we do, but we also care of how we do it. Equity and diversity is at the foremost. It's crucial for our approach to scientists. There is great credit to young scientists, and scientists are leading the initiative. The HCA is organized in biological networks. There are groups that work on specific tissues and organs, and their objective is to answer specific questions. My role within the HCA is to lead the heart biological networks, to co-lead with two colleagues, Rai and Norbert, in the US and Germany, respectively. What is the impact of the HCA? I mentioned it before. The HCA will drive us by creating a reference atlas, first of all, of healthy tissues and organs. We will have our control to make a lot of inferences and comparison with all the disease. We can't study the disease if we don't know what's normal. So it will guide us to understand disease mechanisms, regenerative biology. It will guide us to find new diagnostic markers, new prognostic markers, and to discover new drugs. What have we been doing with the heart? The heart is a large organ. It's probably as big as your fist, okay? And it contains different structures with specialized functions. So we needed a big team. We are now working with a number of scientists, and 4 labs are leading on assembling the Human Reference Cell Atlas. We're creating a consortium. We are working together. The leading labs are the Siteman labs in Boston at Harvard, my lab here at Imperial, the lab of Norbert Hubner in Berlin at the MDC, and the group of Sarah Teichmann in Cambridge. The second element that is fundamental to create a heart cell atlas is the donors. We need human tissue. So we are extremely grateful and thankful to the patients, the donors, and their families that have donated the tissue to allow us to understand better the heart. And of course, you need money. So we're very thankful to all the funders that are supporting us in these studies. So single-cell genomics are helping us to address a number of unanswered questions about the cardiac cellular landscape. This slide is a little complex, and I will not go through the details, but what I want to tell you that for each cellular compartment of the heart, we have a number of questions that need to be addressed. That's the same for the cardiac muscle cells or for the cells of the blood vessels. We need to understand their heterogeneity. We don't think, and we know now that the cardiac muscle cells are not all the same within the heart, but they have slightly different functions. Likewise, for the endothelial cells, the cells that line the inside of blood vessels in the artery versus a vein are quite different. So how did we set up the experiments? Well, we started, as I said, with healthy donor hearts, and we focused on 6 different anatomical regions. We decided to study first the right and left atria, right ventricle and left ventricle. The interventricular septum in purple is the septum that divides the two ventricles, and the tip of the heart, the apex. We studied 14 hearts, 7 adult hearts, 7 from women and 7 from males. But here we are. In science is normal. You always have a challenge. And we did have a specific challenge for the human adult heart. The cardiac muscle cells are very large cells. Well, the length is about 100 microns, and that's equivalent of the thickness of your hair. You might think that's small, but in terms of cells, that's a large cell. I have a cartoon here of a cardiac muscle cell versus a lymphocyte, a small cell of the blood, and you can see the difference in size. The cardiomyocytes won't go through the instrument that allow us to capture single cells. It will simply clog it. The second problem is, or challenge, is that some cardiomyocytes have actually 2 nuclei. So what can we do? Well, we decided to use a combined approach, to use 2 approaches to make sure that we capture all the cells of the human heart. We always had pieces of the heart from the 6 different anatomical regions. So one portion of that was dissociated, as I mentioned before. We get single cells. We get rid of the big cells, which are the cardiac muscle cells that otherwise will clog our instrument. So we get a really good idea, good capture of the non-cardiomyocyte fraction. And then on the other side, we decided to, we are able now to isolate a part of the cell, which is the nuclei. The nuclei is the contains the transcriptional machinery of the cells. In other words, that's exactly where the cell is deciding which genes to activate. So it will contain a lot of crucial information about the ID card of the cell. Here I'm showing this map. I found it amazing. I hope you do the same. You have no idea how much information is packed into this map. We're showing here data from almost 500,000 single cell from 14 human hearts. Each dot here, you can barely see them, represent 1 cell from the heart with its own ID card. So the number of genes that is expressed in these cells will determine where the cell falls into this map. And cells with a similar signature will form clusters. We have identified 10 main cell states, and now we're identifying even more subtypes that we didn't know about. But I won't go into those details. What I wanted to show you that our experimental setup worked well. And then by sequencing single nuclei, so a fraction that is smaller of a cell, we could capture the cardiomyocyte. And we can now gain a lot of information about this crucial cell. How can we use this data? Well, we can use them to make inferences on the cell functions that we didn't know about and have at a much higher resolution. We can drive novel hypotheses, and we can address specific questions. Here we are indeed. The beginning of this year, we all know COVID-19 started to emerge. And COVID-19, we know, is a disease that can present with a variety of symptoms in patients. Some patients that are infected by the coronavirus will be asymptomatic. And we go to the other extreme with patients that can die and will die because of this disease. The common symptoms involve the airways, but many other organs can fail in severe cases of COVID-19, including the heart. That's how we started to, we started to use some of our data to understand if we could help understanding and progressing the understanding of, COVID-19. So how could the Healthy Reference Atlas inform disease? We need to go and look how the virus works. So this is a 3D render of a coronavirus. Can you see all of these spikes here? These are crucial structure in this virus. Basically, the virus uses the spike like a key to enter into the cell. Of course, a key needs a lock on human cells. And the lock on human cell is a specific receptors identified by colleagues in Germany as ACE2 receptors. So the spike protein engages the receptor, and then there is an enzyme that comes in, cleaves off the spike so the virus can enter the cell. What does this all mean? It means that the presence of the receptor of ACE2 is critical to predict in which cells can be infected by the virus. So an even larger group of scientists got together, mostly scientists from the Human Cell Atlas, and we decided to map the expression of ACE2 in healthy tissues. We came together, 80 of us from all over the world, several countries in Europe, in the States, and we all pulled together data. There was the scientists that were studying the airways, our cells with the heart, and up to 20 tissues. So this slide that I'm showing now is very complex, and I don't expect you to read all these names and what cell types we have analyzed. I think it suffices to say that we have surveyed 20 tissues. Because of single-cell genomics, we could exactly pinpoint which cells are expressing the receptor and therefore are potential target for the virus. I'm giving you a couple of examples. In the mucosa of the nose, we have identified that the receptor and the enzyme necessary for the virus to enter into the cells is expressed in two specific subsets of cells. In secretory cells that produce the mucus in the nose, and the ciliated cells that have these little fringes that wipe away particles from the epithelium. In the heart, and that's the data sets that the heart team contributed, the receptor is expressed of a number of cells, including fibroblasts that I mentioned before, as well as these cells that are called pericytes. It's a subset of cell expressed on vessels or the microcirculation on capillaries in the heart. So now the next set of experiment that we're doing as a scientific community, my lab is doing it, people from the HCA and in the world is starting to analyze tissues from diseased patients at various stages of disease. Some colleagues are looking at patients that just arrived, tissues from patients that just arrived in A&E. Other people will look at diseased patients, tissues from diseased patients. So it's fundamental that the scientists, the clinicians, and the patients, that everyone works together to try and make sure we've got tissue available and that we use all the possible information to further our understanding of COVID-19. So thank you so much. I hope you enjoyed this sneak peek on the marvelous diversity of the human heart cellular landscape. And now I will give the floor to Sarah, Professor Rankin.
Great. Thank you so much, Michela. That's, I mean, really fascinating. And, you know, it's interesting because this sort of technology, as you say, this has only become available in the last couple of years. So this is sort of really cutting edge stuff. Before we could only look at sort of genome, as you say, of bulk tissues and compare the whole heart, for example, with the whole liver. So it's incredible the sort of level of detail that we are getting to. And I think, you know, you make another really critical point in that although we're the scientists, none of this work is possible without people being prepared to donate tissue. And, you know, recognizing that people are doing that under sort of really difficult circumstances when they're obviously ill, etc. So I think, yeah, we really acknowledge that and recognize that. It's a really important thing. And, you know, that's how science moves on if we have clinical trials or anything. it's interesting with those spike proteins, because we've seen all that image on the BBC, and I know those or sorry, shouldn't say that, but any TV and the media, and yeah, I didn't realise that they needed to be cleaved with an enzyme to allow entry of the virus into cells. And I guess with my pharmacologist hat on, I'm thinking, does that mean that there's a drug that could inhibit that enzyme, and would that prevent the virus infecting the cells? So maybe we're going to hear about the drugs and pharmacology in a couple of weeks' time. And as you've heard on the news today, we've been there's sort of progress in repurposing old drugs to use this to treat this disease. So yeah, all this science is happening very rapidly in front of our eyes, and for scientists, I think it's really it's fantastic to be a part of this and to see how quickly our research is being translated into sort of benefit for the general public and patients and everyone. So it's really amazing, because this is really why we do science, because we want to make a difference. We want to make the world a better place, I guess.
Absolutely. So, everyone got together very fast. There is a very nice way we share data, and very fast. It's absolutely crucial that we understand that.
Yeah, and I think I think one of the things, certainly my experience of science is that when you go to we have science you do on a global stage, you publish your results, everybody all across the world reads them, and you interact, you collaborate with people from all across the globe. You go to, well, we used to go to conferences and meet up with, for example, like go to a big conference all about heart disease or whatever your sort of specialty is. And the sort of politics, doesn't seem to come into it. It is about the science and furthering science. And I think that's one of the reasons, it's one of the things that I enjoy about science.
Absolutely.
Anyway. Yeah. So please keep posting your questions if you have any for Michela. And to sort of finish this off, I thought you might like to hear from a couple of students associated with Michela's group. So first of all, I'd like to introduce you to Elsa Lawrence. Elsa's currently doing her research project for her master's in Michela's group. So Elsa, can you tell us, well, first of all, how you ended up studying for this master's and what your research project actually is?
Yeah, of course. Hi guys, so I'm Elsa. I went to sixth form in Canterbury in Kent, and for my A-levels I did physics, maths, biology, and also wrote a scientific dissertation for an EPQ, which is an extended project qualification. Some schools do it, some schools don't. So, and I knew that I wanted to go into science, but I was one of those people that wasn't like completely sure or like what I wanted to do at uni. But I really enjoyed biology and I was good at it. So I applied for biology, biological sciences at Imperial. So yeah, I spent 3 years doing my BSc at Imperial. And one of my final year modules was stem cells and regenerative medicine and aging. And I really, really enjoyed that module. That was my favourite module. So I decided that I want to carry on with it in the future. So yeah, that's why I applied to this master's at Imperial. I wanted to stay at Imperial because I've made some really, really good friends here. So it was just really convenient. And also I wanted to definitely be in London. London is like a fantastic place to be as a student. There's just so much to do. And yeah, also there are some really, really great societies at Imperial. So I was part of the Women's Football Society, during my final year, and I wasn't quite ready to say goodbye to that yet. So yeah, I got to play football for another year. But on to my master's. So now, yeah, I'm doing my project with Michela, and I'm doing some computational analyses of single-cell RNA-seq data from mice. And so going back to the 10 main cell types in the heart that Michela was talking about. So for my project, I'm focusing mainly on the cardiac fibroblasts. And I'm investigating whether and if there are any, and if so, like what kind of changes there are in cardiac fibroblast cells after ischemia, so after a heart attack. So yeah, that's my project.
Great. Yeah, I didn't know you played football, Elsa. That's really interesting. Doing that obviously alongside your studies. So interesting that you're using a lot of computational techniques, because we're finding that with a lot of the talks that we're hearing. I mean, obviously, you know, we're using that computational capacity is really important, and allows us to analyze stuff much faster.
Yeah, definitely.
So, right. I'd now like to introduce you to Susanna Jablonska. Susanna is currently doing her master's again in genes, drugs and stem cells, and in October she's going to be starting a PhD actually working in Michela's group. And Susanna, can you tell us again, you know, how did you end up at Imperial? Because I know you had a slightly different transition, and what exactly is a PhD? If you can tell people about that and what you're going to be studying during your PhD.
Yes, thank you, Sarah, for the introduction. Hi, my name is Zuzanna, and I'm an international student, originally from Poland. That's where I did my bachelor's in biology, and that's essentially where my whole science journey began. So then I decided to apply to Imperial to do my master's in genes, drugs, and stem cells. Because I've always been interested in developing novel therapies in medicine, and doing that from the more scientific point than being an actual medical doctor. So I've been fortunate enough to get the Dean's Scholarship, which allows me to undertake this degree being fully funded. Obviously, Imperial is a great institution. They offer a lot of clubs and societies, as Elsa mentioned before. I myself am a member of the Women in Science, Engineering and Technology Society, which does a lot of networking activities to help girls and women in technological and scientific degrees, careers, find their way and get mentorship. And I'm also a member of the Cross Country and Athletics Club. And as Sarah mentioned, in October, I will be starting my PhD in cardiovascular research under the supervision of Michela, as well as Ed Tate from the Department of Chemistry. And a PhD is essentially a fully research-based degree where a candidate is fully funded. Just like me, I'm funded by the British Heart Foundation for 3 or 4 years to undertake their own research to answer a new original question. And in my PhD, I will be focusing on finding a new way of preventing cell loss after a heart attack.
Thank you, Susanna. Yeah, there's a lot of interdisciplinary work going on at Imperial. And for example, in my lab at the moment, I've got a physicist, I've got material scientists, and I've even got artists that I'm working with. So that's all good fun. And I guess one of the things that I really love about being a scientist is that you continue learning new things all the time. So you never get a chance to get bored. Right, Margarita, how are we doing? Are there any questions from people watching?
Yeah, hi Sarah.
Hiya.
Yeah, we have plenty of interesting questions this time.
Okay,
I will start with Michela because I think she's the best in that. So the first question is from Jade. Jade is asking, does the virus prefer certain cells expressing ACE2 receptors, or does it infect all the cells with this receptor?
That's an excellent question, Jade. The short answer is we don't know yet. And that's why it's fundamental for us to study now the diseased tissues. This way we'll be able to say and to see and exactly map where the virus has entered. I'm working in collaboration with Omer at the Sanger Institute and we're running experiments on autopsy tissues to map exactly where the virus is present, in which cell types. And then we're going to do single cell genomics on the cells that have been infected by the virus versus cells that haven't. These will hopefully guide us to understand why some cells will be infected or not. Keep in mind that the receptor based on in vitro studies is likely not sufficient. You also need the enzyme that I've shown you before. There are 2 extra candidate enzymes that might help the virus as well. So the situation is obviously more complex than we think, and we have so much to learn still.
That's great. So we have another question asked the same from Roshan Patel. So his question is, since children have a lower risk of dying due to coronavirus, could it be due to a difference in ACE2 receptors or on cells? Could they not have developed as mass or not matured enough?
That's another excellent question. So we need to do more to analyze the expression of the ACE2 receptors on tissues from children. What I can tell you, for example, that there are different levels of expression in males versus females in the adults, with ACE2 receptor being higher level in males, and that might justify in part while we know that men on average have are more seriously affected than women. And again, just imagine how, from our scientist's point of view, we know so little. It's been just, well, few months that we have started to try and understand, because we didn't even know this disease existed. So every day we're discovering something new. But your question is absolutely good. And I know exactly colleagues that are looking specifically at tissues from children.
Great. So another question for you, Michela, is from Alice. And she's wondering at which stage is drug development for COVID-19 in the UK currently? Do you know that?
I would, well, the question is good, and it's also very broad. There has been a lot of effort in investing for a vaccine, and I do think that maybe there has been less oomph on thinking and investment of drugs that will or can block the viral infection. There has been, and there is a lot of thinking of how we can repurpose existing drugs that might work for the virus. But I want to underline again, we're so early in understanding the virus that it's very hard. But I will maybe pass to Sarah this question, who's a pharmacologist, and maybe can add something more.
Yeah, I mean, I think on the 30th of June, we're going to have a whole session on this and drugs. And I think essentially the answer to your question is that we needed something to treat patients now. And normally it takes at least 10 years to develop a drug. So, for example, if we had decided even last month, if we thought, okay, let's develop a drug that will inhibit that enzyme and stop COVID-19 infecting cells, even if we had developed a drug, that process of developing it, putting it through all the sort of testing that you have to do in animals and in humans, takes about 10 years and cost a huge amount. And so that wasn't seen as a sort of relevant way of going forward in the sort of first instance. And so what people have been doing is looking at, well, what drugs have we got on the shelf at the moment that could affect this? And one of the drugs is a steroid called dexamethasone. That's the one we've been hearing about today. We've also heard about, there's another drug that was used to, when they had the Ebola virus, the epidemic, that was used in that case, and that one has been showing some effect. What has been important and what we have been able to do in the UK, we have been very fast at organising and running clinical trials for these drugs so that we can actually test whether they are actually being effective. And we haven't yet seen the data for the statements that are being made at the moment in the media about the dexamethasone. As scientists, as I haven't yet had an opportunity to look at the data that supports those claims yet. But at the moment, it sounds like for certain groups of patients, the ones on ventilators, this could be a useful drug. So this is what we mean by repurposing a drug, using a drug, and that drug gets used for patients that have other respiratory and inflammatory diseases, for example, like asthma or COPD. So yeah, interesting.
Thank you, Sarah. So we have another question, the last but not the least, for Michela. So Chris is asking, does CRISPR-Cas9 help with single-cell genomics?
That's a very, very clever question. I think I like it a lot. So I would say that the integration of CRISPR and Cas9 with single-cell genomics can help us a lot, definitely. It's keeping in mind that with CRISPR-Cas9, we need to know what usually when we use it, because we have an hypothesis-driven approach. In other words, if we think a gene might be relevant, for example, for inducing proliferation of a certain cell type, for example, for cardiomyocytes to re-enter the cell cycle, this would be it would be crucial to put together the two technologies using in vitro system to see whether when you lose that gene of interest, you can, for example, block the proliferation of the cells, or vice versa, induce. So I would say that it's an absolutely good option to put together to make, design validation experiments, functional validation experiments in an hypothesis-driven approach.
Great. So I think this topic has sparked a controversy because there are more and more questions coming. So Chiara has a clever question. So she's wondering, has there been research into if the immune response to COVID-19 is more focused on the use of T cells or B cells? Is the response humoral or cell-mediated? Sorry.
I can answer. Well, I think what Chiara, maybe have a look at the first of our COVID lessons, because we had Danny Altman, the professor of immunology, telling us about the immunological response to COVID-19. So yeah, I'd refer you back to having a listen in on that lecture to hear, and definitely we are investigating. Immunologists are definitely looking at that. What is the type of immunological response?
Good. So another question, Joella said that there have been reports about certain blood types that are more susceptible to COVID-19. Do you think this is significant, and why do you think it's happening?
Yeah, I know that's not Michela's area of expertise or particularly mine, so I haven't read that actually, Joanna. So I think I have seen it mentioned, but I haven't read further about the scientific basis for that. So sorry, can't really comment on that one.
That's okay. We can learn a lot of things during this session. So I think in the next lockdown lessons, we will have the opportunity to learn something about it, because they have to stay tuned and see all the next and upcoming lessons. Yeah, not forget to vote for that and to be ready for it. So I want to ask a question to Elsa and to Susanna. As there are a lot of people out there who are wondering how is the life of a student at Imperial in London. So Elsa, do you want to come first and answer me?
Yeah, sure. Was the question what's my favorite thing about Imperial?
Yep.
Yeah, that's difficult. I'd say, yeah, personally, I'd say I love meeting different people from different backgrounds, and like there's a lot of diversity at Imperial. So I think that's one of the favorite things about Imperial for me, was just meeting so many people during my undergrad and for my masters. So yeah.
Yeah, that's one of the things that I love as well. I'm just butting in. I'm not a student, obviously, but I love having, I love teaching the students here, because we do have a whole range of students, and just really interesting on our master's course, which, although it's genes, drugs and stem cells, we've had people that have done degrees in economics or law, and they're interested and they come and do our master's course. And I think, yeah, there's a real sort of range of students from all different backgrounds, international students, obviously we have them from, and I think that really makes, when we have discussions about the ethics and the science, we're really thinking on a global level, because we can get the views of people that have come from all different parts of the world, and I think that really enriches the sorts of discussions and activities that we do.
Susanna, do you want to make any comment about it?
Yeah, sure. I definitely agree with Elsa and Sarah that the diversity is a great part of being at Imperial. And I would also mention that the multidisciplinary research and the collaborations between teams from various groups, as we already mentioned before, is something that really, as a young scientist, is very, very inspiring, that we can talk about our research, about other people's research with so many different people, with experts in their fields, and attend their lectures and tutorials. So I think that is a great thing about Imperial.
Thank you, Susanna. That's all our questions.
What about you, Margarita? Because you've been here as well, and you're coming from Greece. What was your sort of what did you think of living in London? And
the first and the major difference is the weather. You know that.
It's pouring with rain today, I have to tell you.
Oh, really? No, here it's really sunny, and there are a lot of people going to the beaches, doing outdoor activities. Because we have been eased from lockdown. Anyway, I think what I loved about London is that you can go everywhere, and it's like the center of the world. If you cannot travel to New York or to United States or to China, whatever, you can meet different cultures and civilization at the same place. And that's really important for me, because I have studied in Greece, so I didn't have that multicultural background. And I really enjoyed meeting people from all over the world and exchanging ideas and scientific projects. That is really important because science has to do with interaction and collaborations. So I think London is a spot for science and for research. That's why Imperial and other prestigious universities are so highly qualified in the rankings.
And it's interesting, this sort of interdisciplinary idea. As I say, I love working with people in other departments at Imperial, and now I've started projects because within where Imperial is in South Kensington, we're in an area that is known locally as Albertopolis. We have the Royal Albert Hall, we have the Royal College of Music, the Royal College of Art, the Science Museum, the Natural History Museum, and the Victoria and Albert Museum. And we work together to run an amazing festival called the Great Exhibition Road Festival once a year. And that's a really fun event. And what we are doing with that event now is to work collaboratively. So the scientists with the musicians or the artists to develop activities. And it's just really cool to do some of those sort of more interesting projects on the side, and you meet these incredible musicians, for example, or get to see behind the scenes in the Victoria and Albert Museum. It's just it's such a sort of rich cultural area. Anyway, I'm going to stop yabbering, and I'd really like to just say thank you to our speakers again. Michela, she always gives such great, clear presentations. Elsa and Susanna, thanks for giving us an insight to being a student, and clearly Margarita, obviously, for moderating the session, and Sam, who we haven't heard or seen from yet, but he's in the background producing the session. So tomorrow we'll be back with some of Imperial's epidemiologists. Now, if you didn't know what an epidemiologist was before COVID-19, I'm sure you do now. They're the people that have been modelling the spread of the virus. And particularly this group, Imperial's epidemiologists, are the people that sort of were stepping in and telling the government to, that they needed to lock down soon to prevent huge numbers of deaths. And so they've been very much not only in doing their science, but also they've been in the news a lot and advising the government. So I've just been, Margarita, you're just sort of telling me there's another question, yes?
Yeah, there is another question from Alice. Yeah, sure. So Alice is asking, do you work with medical doctors when researching or studying at Imperial?
Definitely. Yes.
Yeah,
very much so. I mean, we are the Heart and Lung Institute. Both Michela and I are in the Heart and Lung Institute, which is part of the Faculty of Medicine. We're associated with the Imperial Academic Healthcare Trust. So this includes St Mary's Hospital, Charing Cross Hospital, and Hammersmith Hospital, and Brompton Hospital. We have, in my department, is respiratory. We have a lot of respiratory physicians, and we are going to be hearing from some of those physicians later in the series. They actually do some of the clinical research, but they also work on, in the actual clinics. And during the lockdown period, they've all gone back full time into hospital work because obviously they were needed on the wards. And we also have these people, I think we might record another session from one of my, I had a medical student that did a PhD with me. He's now working in Glasgow in the intensive care unit. But yeah, so we very much, because as I say, for us, sometimes the clinical people are the people that really can give us an insight into the sort of nuances of disease processes, because they obviously are experts in their own specific areas, and they also allow us to interact with patients, for some of our studies. So yeah, we're very closely aligned with clinical people, and we train all the medical students. So yeah, for many years I've taught pharmacology to the medical students.
So this is it.
Right, that's it. That was
Yep.
the last sort of question sneaked in there by. Anyway, so thank you all for joining us. Thank you for those that have been watching in. Please fill out the survey that should be in the chat now. I think Sam is going to put it there. It's a very quick survey, but it just, you know, gives feedback to these guys that have spent their time developing the webinars for you. Enjoy the rest of your day, and I hope to catch up with you again tomorrow, where we're going to be hearing about the modelling the spread of the virus. So goodbye from all of us. Bye.
Bye.
Bye, guys.